Hi everyone, my name is Shelby and I am a graduate student at the University of Alaska Fairbanks. And today I'm going to be presenting my thesis, which is the effects of ocean warming and ocean acidification on the behavior and physiology of an intertidal grazer and a key sea star predator. But before I begin, I just want to say a big thank you to my funding and project partners. This project was funded by Alaska NSF EPSCoR and the Robert and Kathleen Byrd Award. So as most of us know or have at least heard, our oceans are in a current state of change. And over the past 100 years, what we've seen is that atmospheric CO2 is steadily increasing. And this is largely driven by anthropogenic forces such as deforestation and the burning of fossil fuels. Additionally, we're seeing an increase in the uptake of oceanic CO2. And this is leading to a concomitant decrease in oceanic pH. Unfortunately, this is most prominent in polar and subpolar regions where CO2 solubilities in water is increased in cooler climates. In addition to decreasing pH, we're also seeing an increase in sea surface temperature. And these increases in temperature and decreases in pH can affect many marine organisms, and it can affect them from the molecular to the organismal biological level, but also can affect populations, communities, and ecosystems via modified species interactions. So in order to understand how our communities and our ecosystems might respond to future ocean change, we first and foremost need to characterize the species' individual vulnerability to ocean change and multiple stressors. Now, in order to do this, we often look to the rocky intertidal ecosystem. And this is because the rocky intertidal is characterized by being a dynamic physical and biotic environment. We see daily fluctuations in things such as temperature and photoavailability. And because of this, the rocky intertidal is defined by this vertical zonation that we see. So vertical banding of organisms in this ecosystem. And the upper limits of a species range is usually set by abiotic factors, so things such as temperature tolerance or desiccation tolerance, whereas the lower limits of a species range is often set by biotic factors such as competition and predation. So the interesting thing about vertical zonation is that it directly reflects a marine organism's adaptation to varying degrees of stress. And this makes it the ideal study system for multiple stressor experiments. Now, our project looked at two species, a predator and a prey relationship, and we started with Lania sputum, which is the Pacific plate limpet, and it's an important intertidal grazer. And due to high predation pressure by seabirds and sea stars, Lania has developed a really interesting set of anti-predator responses and behaviors. It can, number one, suction down onto rocky substrate at tremendous forces to avoid being pried off. It can also run away or flee from its predator if it comes in contact with waterborne or tactile cues. And lastly, in close encounters, it has the ability to wiggle its shell side to side or rotate it completely around its visceral mass to avoid the grasp of a close sea star. Now, our sea star predator in this experiment was Evisceris trocheli, or the mottled star. And the mottled star and Lania have significant range overlap, both geographically and at depth. And because of this, it's provided us with a very well documented predator-prey relationship in the literature in Alaska's nearshore ecosystem. And because it's a dynamic predator and prey relationship, it's the perfect model to characterize the effects of ocean change, not only on individual species, but on their interactions with each other. So this project looked to determine the individual and interactive effects of ocean acidification and ocean warming on 4 different parameters. One being the anti-predator behavior of Lania in the presence of its sea star predator. So whether or not behavior is modified by OA and OW conditions. Number 2, we wanted to look at the physiology of Lania. And to do this, we looked at metabolic rate and thermal tolerance. Number 3, we wanted to look at the stress response of Lania both in OA and OW conditions, but also in the presence of their sea star predator. And to do this, we look at cortisol, which is a stress hormone that is expressed when organisms experience and undergo stressful conditions. And lastly, we wanted to look at the neural response of Lania and whether or not behavior is modified because of neurotransmission lapses or miscommunication in the firing of neurotransmitters. So we can look at this by looking at GABA receptors. Now, our project took place at the joint NOAA and UAF Kitsitsa Bay Laboratory from July through August 2020. And we collected individuals from adjacent rocky intertidal habitats and allowed them to acclimate to the laboratory environment for 7 days. After acclimation, they were randomly divided between 4 different treatments. And each treatment consisted of 5 individual limpet culture vessels with 30 limpets in each culture vessel, and 1 C-star culture vessel with 1 C-star in each culture vessel. Our ambient temperature and elevated temperature for this experiment were 11 degrees Celsius and 15 degrees Celsius, and our ambient pH and decreased pH were 8.0 and 7.6. Now, ambient temperature and ambient pH for this experiment were calculated directly from incoming seawater at the lab, and elevated temperature and decreased pH were calculated based on the IPCC's projections for the year 2100, based on the business as usual category. This experiment was 29 days long, and temperature and pH were all maintained via the flow-through ocean change experimental system, or OSIS for short. Over the 29 days, we conducted multiple experiments, starting with numerous oxygen consumption assays, which we later can use to calculate the metabolic rate of Lania. We also conducted a thermal tolerance assay on days 14 and 15, and we collected numerous tissue samples so that we could later calculate cortisol levels in these samples at our laboratory here at the UAF main campus. And lastly, on days 21, 28, and 29, we conducted 3 different behavior experiments. Now, we do have preliminary results, and we'll start with our thermal tolerance experiments. And what we can see by the figure on right is that we have our CT max on the y-axis, and CT max is the critical thermal maximum of Lania, or the temperature at which they expire. On our x-axis, we have our 4 different treatments, and on our plane, we have points which represent the mean CT max for each treatment, and error bars which represent the standard deviation. Now, using a two-way ANOVA, we analyzed our results and found that temperature had a significant effect on the upper thermal tolerance value of Lania. So the temperature at which each treatment was acclimated at, whether that being 15 degrees Celsius or 11 degrees Celsius, significantly affected the CT max of those treatments. However, we found that pH did not have a significant effect on CT max, and that there weren't any interactive effects of pH and temperature. Now, metabolism was also interesting, and we ran a 4-way ANOVA for this, and we found that there were no significant effects of temperature, pH, or day/time on the metabolic rate of Lania. However, we did find that there was a significant effect of treatment on the metabolic rate of Lania. And we can see this in the figure on right. So on our y-axis, we have our metabolic rate standardized to size. And on our x-axis, we have our different treatments, including our day 0 field caught Lania, so Lania coming directly from the rocky intertidal ecosystem. We have our lab acclimated Lania and all of our treatment conditions. And what I want to point out is that the field caught day 0 Lania have a significantly higher metabolic rate than any of the lab acclimated and treatment conditions. Lastly, we found that there weren't any interactive effects of pH and temperature on the metabolic rate of Lania. Now, over the next few months, we are going to wrap up the data analysis of this experiment. Concluding with our behavior experiments, we will measure path length, amount of time spent out of water, path shape, and direction of rotation to determine whether there was any behavioral modification in Lania. And we will also begin to measure cortisol levels in the tissue samples that we collected over the course of the experiment. Now, before I go, I just want to leave us with some thoughts on the implications from the study and the results that we have so far. So looking at thermal tolerance and metabolism. So we found that acclimation to a higher temperature actually decreased the overall thermal tolerance of Lania, which is quite interesting because it's not what we usually see in these kinds of experiments. Usually when organisms are acclimated to a higher temperature, they express a higher CT max. However, we see the opposite results. And what this could mean is that higher acclimation temperatures are inducing a high toll or a high physiological stress, enough so that CT max is lowered. And this is significant because it means that species, even when acclimated to warmer conditions, are sensitive to OW. Additionally, we found that field caught metabolic rates of Lania are higher than the laboratory metabolic rates. And this is interesting, and again, this could mean that the laboratory environment and condition is inducing physiological stress, and Lania is responding by decreasing its metabolic rate to meet the energy deficit demand to maintain homeostasis in their body. So with that, I just want to say thank you so much again to my project and funding partners. I want to say a big thank you to my advisor, Dr. Amanda Kelly, and my committee members, Sarah Hardy and Brian Gaylord. And lastly, I want to thank my lab mates, Marina, Jamie, and Max, all of which this experiment could not have happened without their help. Thank you so much. And with that, I'll take any questions.